to Kalamazoo College, which is a really small liberal arts college in Michigan. And I thought I wanted to do pre-med. And then after taking some intro chemistry courses, my organic chemistry professor convinced me to go try doing research in a lab. That kind of switched my whole focus towards research. I did an internship at Eli Lilly in their in vitro drug metabolism and toxicity group. And so that was a really cool experience where I was able to see what research was like in industry, which was a lot different than my small liberal arts college. It was a really awesome experience and kind of pushed me that I do want to do some type of drug development related research, and that I did also want to attend grad school and continue to do research and get my PhD. I started at Northwestern. And I joined Gabe Rockland's lab in the pharmacology department here down at the medical school campus. The goal of our lab overall is trying to learn how we can make better protein drugs for treating different types of disease. Once people are making protein drugs, they have to go through like manufacturing, transportation, the formulation has to be designed and then be stored and then get into the body. And during all of these processes, there could be different stresses imposed on the drug. So, the temperature might change, the pH might change. It might have to get to a really high level of protein concentration in the formulation. And that any of those things can cause the protein to change its shape. And then when that happens, they can clump together. These clumps are called aggregates. And it's really important to try to reduce this process of aggregation, because these clumped proteins that are no longer have the shape you wanted them to have, they can cause your body to react negatively and have immunogenic effects, which really can reduce the safety. They also don't have the same shape they were designed to have, so they might not interact with what you wanted them to, and that can really reduce the efficacy as well. And so right now it's kind of challenging to know how well a protein therapeutic may or may not aggregate and how well it will do under those types of stresses. So my work is kind of trying to get at that question. So what we really want to do is develop a method where we can measure how much a protein will aggregate when it is exposed to any of those stresses, and then do this for thousands of proteins at a time. And so by generating like this really large amount of data, we can actually start to learn like why are some proteins aggregating and some aren't, which is hard to do when you only are studying a few proteins at a time. And once we can learn general trends like that, we can then use those lessons to say, how can we make, if we want to make a desired drug, how can we take those lessons and incorporate it into it so that drug might be less prone to aggregating? Or can we make a model that we could actually put a bunch of candidates in and say, these are probably going to perform better than these. Kind of twofold: one in giving me more confidence in pursuing a research career into some type of research on protein therapeutics, but also like increasing my network with people that are in the PhRMA Foundation, other fellowship winners, and other like career talks and connections that the PhRMA Foundation provides.